Hello children, welcome for today's crack NEET biology classes. So today we are going to learn MCQs from the chapter Cell, the Unit of Life. So first MCQs will be read for you. You try to find out the answer. If you don't know the answer, answers will be given followed by the explanation. If you want more explanation, you can go to the description box. It has been given there. And if you want NCERT explanation and entire chapter, that also I explained and attached in the description box. You can go through that also. Now let us start the session with a motivational thought, children. Don't watch the clock. Do what it does. Keep going. Like a clock, continuously you have to work to get good results. Now let us start our session. First question. A major breakthrough in the studies of cells came with the development of electron microscope. This is because A. Electron microscope is more powerful than the light microscope as it uses a beam of electrons which has wavelength much longer than that of the protons. B. The resolving power of electron microscope is much higher than that of light microscope. C. Resolving power of the electron microscope is 200 to 350 nanometer as compared to 0.1 to 0.2 nanometer for the light microscope. D. Electron beam can pass through thick materials whereas light microscopy requires thin sections. So now let us find the correct answer. B is the correct answer. Why it is correct answer? Let us see. Microscopes are used for studying cellular structures. They are used to magnify small objects. In electron microscope, a high energy beam of electrons is focused through electromagnetic lenses. It can magnify very small details with high resolving power. Now let us go to the second question. A student wishes to study the cell structure under a light microscope having 10x eyepiece and 45x objective. He should illuminate the object by which one of the following colors of light so as to get the best possible resolution. A. Blue, B. Green, C. Yellow, D. Red. Now the correct answer is A. Blue. The resolution power of microscope is inversely proportional to wavelength of light used. Out of 4 options given, blue light has minimum wavelength and hence maximum resolution. Now let us go to the next question. The electron microscope has a high resolution power. This is due to A. Electromagnetic lenses, B. Very low wavelength of electron beam, C. Low wavelength of light source used, D. High numerical aperture of glass lenses used. The correct answer is B. Very low wavelength of electron beam. Here microscopes are used for studying cellular structures. They are used to magnify small objects. In electron microscopes, a high energy beam of electrons is focused through electromagnetic lenses. It can magnify very small details with high resolving power. The increased resolution results from the shorter wavelength of electron beam. Now let us go to the fourth question. Magnification of compound microscope is not connected with A. Numerical aperture, B. Focal length of objective, C. Focal length of eyepiece, D. Tube length. The correct answer is A. Numerical aperture. The magnification of compound microscope is not connected with numerical aperture. The magnifying power is represented by the symbol X. You must have seen in the lenses. The total magnification of a microscope is obtained by multiplying the objective power magnification and ocular lens magnification. That is, if the magnification power of an ocular lens is 10x and object is of 40x, then the total magnification power of the microscope is 10 multiplied by 40 equal to 400x. Now next question. The concept of omnis cellulae regarding cell division was first proposed by A. Aristotle, B. Rudolf Virchow, C. Theodor Schwann, D. Schleiden. The correct answer is B. Rudolf Virchow. So Rudolf Virchow in 1855 observed that new cells develop by division of pre-existing cells. His finding gave cell theory to the final shape. Now let us go to the next question. Which is correct about cell theory in view of current status of our knowledge about cell structure? A. It needs modification due to discovery of subcellular structures like chloroplast and mitochondria. B. Modified cell theory means that all living beings are composed of cells capable of reproducing. C. Cell theory does not hold good because all living cells, example viruses, do not have cellular organization. D. Cell theory means that all living objects consist of cells whether or not capable of reproducing. Now the correct answer is C. C is the correct answer. Viruses are an exception to the cell theory as they are obligate parasites. Now the next question. Names of Schleiden and Schwann are associated with A. Protoplasm as physical basis of life, B. Cell theory, C. Theory of cell lineage, D. Nucleus functions as control center of cell. Now the correct answer is B. Cell theory. Names of Schleiden and Schwann were associated with cell theory in 1839. The concept that all living organisms are composed of cells is known as cell theory. Now the eighth question. Angstrom is equal to A. 0.01 millimeter, B. 0.001 millimeter, C. 0.0001 millimeter, D. 0.00001 millimeter. The correct answer is C. C is the correct answer. And angstrom, that is the symbol, what is given. It is a non-SI unit of length that is internationally recognized, equal to 0.1 nanometer. So here it is named after Hans Jonas Angstrom. Angstrom equal to 0.0001 millimeter. That's the correct answer. Now let us go to the next question. Organelles can be separated from cell homogenate through A. Chromatography, B. X-rays diffraction, C. Differential centrifugation, D. Auto-radiography. The correct answer is C. Differential centrifugation. So organelles can be separated from cell homogenate through differential centrifugation. The basic principle involved here is sedimentation of particles in a suspension by centrifugal force. In a centrifuge, the particle sediments at different rates when an accelerating force is subjected. The rate of sedimentation depends upon the size of particles, its shape and density. The 10th question. So which of the following statements about inclusion bodies is incorrect? A. They are not bound by any membrane. B. These are involved in ingestion of food particles. C. They lie free in the cytoplasm. D. These represent reserve material in cytoplasm. The correct answer is B. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. So these are not bound by any membrane system and lie free in the cytoplasm. Example, phosphate granules, cyanophycean granules and glycogen granules. Now let us go to the next question. Select the wrong statement. A. Bacterial cell walls is made up of peptidoglycan. B. Pili and fimbriae are mainly involved in motility of bacterial cells. C. Cyanobacteria lack flagellated cells. D. Mycoplasma is a wall-less microorganism. Let us see the correct answer. B is the correct answer. Pili and fimbriae are bacterial appendages which are not involved in locomotion. Next question. So, which of the following structures is not found in a prokaryotic cell? A. Mesosome. B. Plasma membrane. C. Nuclear envelope. D. Ribosome. The correct answer is C. Nuclear envelope. A prokaryotic cell is characterized by the absence of an organized nucleus and membrane-bound cell organelles. Here DNA is naked, that is without a nuclear envelope, and lies in the cytoplasm. The place where the chromosome lies, you call that as nucleoid. Mesosomes, plasma membranes, and 70S ribosomes are present in a prokaryotic cell. So C is the correct answer. Now let us go to the next one. The structures that help some bacteria to attach to rocks and or host tissues are A. Mesosomes, B. Holdfast, C. Rhizoids, D. Fimbriae. The correct answer is D. Fimbriae. Now next question. So which one of the following is not an inclusion body found in prokaryotes? A. Glycogen granule, B. Polysome, C. Phosphate granule, D. Cyanophycean granule. The correct answer is polysome. Polysome is not an inclusion body. It is an aggregation of ribosomes. Now the next question. So which structures perform the function of mitochondria in bacteria? A. Nucleoid, B. Ribosomes, C. Cell wall, D. Mesosomes. The correct answer is D. Mesosomes. Mesosome is characteristic specialization of bacterial cell membrane that develops as an ingrowth. It consists of vesicles, tubules, and lamellae. They also increase the surface area of plasma membrane. Now next question. The motile bacteria are able to move by A. Fimbriae, B. Flagella, C. Cilia, D. Pili. The correct answer is B. Flagella. Flagellum is the organ of motility in bacteria. They perform rotatory movements in locomotion. Now 17th question. Why is a capsule advantageous to the bacterium? It protects the bacterium from desiccation. B. It provides means of locomotion. C. It allows bacterium to hide from host immune system. D. It allows the bacterium to attach to the surface. The correct answer is A, C and D. The S type of bacteria or virulent bacteria are capsulated. The capsule is made of polysaccharides and amino acids. It is a tough and thick mucilage covering. It gives protection to the bacteria against host immune system. The capsule is considered a virulence factor because it enhances the ability of bacteria to cause disease. Capsule contains water and mucilage, thus protects bacteria against desiccation. Capsule also helps the bacterium to adhere to the surface. Now 18th question. The term glycocalyx is used for A. A layer present between cell wall and membrane of bacteria. B. Cell wall of bacteria. C. Bacterial cell glyco-engineered to possesses N-glycosylated proteins. D. A layer surrounding the cell wall of bacteria. The correct answer is D is the correct answer. Glycocalyx is a sticky gelatinous material that collects outside the cell wall of bacteria to form an additional surface layer. So when this layer is firmly attached to the surface of the cell, it is called a capsule. If it is loosely distributed around the cell, no, the glycocalyx is called a slime layer. Now the next question. Chromosomes in a bacterial cell can be 1 to 3 in number, and A are always circular, B are always linear, C it can be either circular or linear but never both within the same cell, D can be circular as well as linear within the same cell. The correct answer is A. A is the correct answer. So the bacterial cells, they do not have nucleus, characteristic of eukaryotic cells. Nuclear material of bacteria lies free in the cell in the form of an irregular, thin, fibrillar and circular single molecule of DNA, and it is located in a place called nucleoid. Okay. In addition to circular DNA, a small amount of subsidiary extrachromosomal DNA is also present as plasmids or episomes. Now next question. Photosynthetic bacteria have pigments in chromoplasts, B chromatophores, C leucoplasts, D chloroplasts. The correct answer is B chromatophores. Photosynthetic bacteria have chromatophores, which are membrane bound vesicular structures which are extension of cytoplasmic membrane. They contain photosynthetic pigments along with enzymes. Now 21st question. The site of respiration in bacteria is A ribosome, B microsome, C episome, D mesosome. The correct answer is B mesosome. Mesosomes are complex intracellular membranous structures within the cytoplasm that are formed by the infoldings of the cytoplasmic membrane. Surface of mesosomes have many enzymes which take part in respiration. Example, the enzymes are oxidases, dehydrogenases are present. They are also called as mitochondria of bacterial cell. Now the next question. So which is the most important sites of formation of glycoproteins and glycolipids in eukaryotic cells? A. Endoplasmic reticulum, B. Peroxisomes, C. Golgi bodies, D. Polysomes. The correct answer is C. Golgi bodies. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Proteins synthesized by the rough endoplasmic reticulum and lipids synthesized by smooth endoplasmic reticulum reach the cisternae of the Golgi apparatus. Here they are combined with the carbohydrates to form glycoproteins and glycolipids. Now the next question. So which of the following statements is not correct? A. Lysosomes are formed by the process of packaging in the endoplasmic reticulum. B. Lysosomes have numerous hydrolytic enzymes. C. Hydrolytic enzymes of lysosomes are active under acidic pH. D. Lysosomes are membrane bound structures. The correct answer is A. Lysosomes are single membrane bound vesicles which contain hydrolytic enzymes. Lysosomes are believed to be formed by the joint activity of endoplasmic reticulum and Golgi apparatus. Precursors of hydrolytic enzymes are synthesized at RER and are transferred to the forming phase of Golgi complex. These precursors then changed to enzymes. Enzymes are then later packed in larger vesicles and are pinched off at maturing phase. These vesicles combine with endosomes to produce lysosomes. Lysosomal enzymes function in acidic medium or acidic pH. So the pH, it can be even they work under 4.5 also. This pH is lower than that of cytoplasm. Now next question. Which of the following statements regarding mitochondria is incorrect? A. Mitochondrial matrix contains single circular DNA molecule and ribosomes. B. Outer membrane is permeable to monomers of carbohydrates, fats and proteins. C. Enzymes of electron transport are embedded in outer membrane. D. Inner membrane is convoluted with infoldings. The correct answer is C. Enzymes of electron transport are embedded in the inner membrane of mitochondria. Now 25th question. The shorter and longer arms of a submetacentric chromosomes are referred to as A. M-arm and N-arm respectively. B. S-arm and I-arm respectively. C. P-arm and Q-arm respectively. D. Q-arm and P-arm respectively. The correct answer is C. The short arm of chromosome is termed as P-arm, and the longer arm is referred as Q-arm. So that's why C is the correct answer. Next 26th question. So which of the following pairs of organelle does not contain DNA? A. Nuclear envelope and mitochondria. B. Mitochondria and lysosomes. C. Chloroplast and vacuoles. D. Lysosomes and vacuoles. The correct answer is D. Now next question. So here match the column. Two columns are given. Match the column 1 with column 2. Then after matching, you choose the correct match pairs as options given below. A, B, C, D, they have given 4 options. After matching, you have to tell the correct answer. The correct answer is A. A is the correct answer. Next 28th question. So which of the following is true for the nucleolus? A. Larger nucleoli are present in dividing cells. B. It is a membrane bound structure. C. It takes part in spindle formation. D. It is the site for active ribosomal RNA synthesis. The correct answer is D. Nucleolus is naked. It does not have any membrane. Round or slightly irregular structure which is attached to the chromatin at a specific region and called nucleolar organizer region. Nucleolus is the site of rRNA synthesis, that is ribosomal RNA synthesis. Nucleolus disappears when the cell undergoes cell division and reappears after its completion. Next 29th question. The Golgi apparatus participates in A. Fatty acid breakdown. B. Formation of secretory vesicles. C. Respiration in bacteria. D. Activation of amino acid. The correct answer is B. Formation of secretory vesicles. Golgi apparatus, Golgi complex, So it is a complex cytoplasmic structure made up of smooth cisternae, a network of tubules with vesicles and vacuoles which takes part in membrane formation, secretion and production of complex biochemicals. One phase of Golgi apparatus is convex, it is forming cis phase, while the other is concave, maturing trans phase. Cis phase receives contents from endoplasmic reticulum, then reaches trans phase and bud off as secretory vesicles. Now 30th question. Which of the following events does not occur in rough endoplasmic reticulum? A. Protein folding. B. Protein glycosylation. C. Cleavage of signal peptide. D. Phospholipid synthesis. The correct answer is D. Phospholipid synthesis. Smooth endoplasmic reticulum is involved in the production of lipids and sterols. Complexing of lipids and proteins to form lipoproteins occur in SER. RER is involved in protein folding, glycosylation of protein. Okay children, hope you must have understood whatever explained today. So revise properly and all the best for your NEET exams. Thank you children. We will meet in the next class.